All right, grade 10s. We're going to cover two things today. One is we're going to go over your first assignment, which is going to be on mitosis. So last time we talked about um, cell division. I think that's what we did last time. So this assignment is in your course outline folder. So here's your course outline folder. All right. So in your course outline folder, just to recap, there is a free online version of the textbook. Here's your grade 10 course outline, all right, with some questions on, the cell division, stuff like that. Um, so the lab is here in lab related, okay? So if I click on that, all right, so here's the lab. This is the document, which I'm gonna go over with you. The lab is based on analyzing a bunch of slides of cells going through either cell division or not going through cell division. These are the slide samples that you're going to look at, okay? So they're all these ones. Now, the lab itself, when you do finish it, in terms of the deadline, I'm going to say 2 weeks from now, all right? And then we'll see how if that needs to be extended or not. The lab is going to go into this folder here. So you're going to submit into this folder. All right. So don't message it. Just put it in this folder. You will have the option to submit. Okay. So make sure you put it in here. If you are sharing a Google Doc, make sure that you allow permission for it to be seen. It's quite annoying when you have to get permission to see it. Okay. So make sure that you have it on anyone can view. All right. So let's go over what this lab is about. It's in relation to what we talked about in terms of cell division. All right, so let's make this just a little bit bigger so we can clearly see it. So if you recall, last time we talked about cells, when they divide, they undergo particular phases. All right. So for instance, if I look at something like this, you can see that the cell in the middle is undergoing anaphase. All right. Um, if I look at, for example, let's see, I thought I had some other slides here. Hold on. Maybe not. Okay. So, for example, something like this would be prophase because you can kind of, shoot, this could be a little bit bigger. You can kind of make out the chromosomes. Let me make this smaller. All right, let's look at this slide. Okay, so for example, this slide here, you can see the chromosomes. So I would say that's prophase. But if I look at this top right corner, I don't see the chromosome. So I would say that that one's, in interphase. So if you remember, the cells are either going to be in interphase, which means they're not actively dividing. They could be at rest, or maybe they're preparing to divide, or they could be going through the stages of mitosis. Like prophase, we talked about metaphase, anaphase, telophase, for example. So, like this one, for instance, this is metaphase. You can see that the chromosomes are lined up in the middle. So your job will be to analyze particular slides, all right? And then answer some questions about the slides. So for example, the first question is about something called a mitotic index, because this is gonna come up in your lab. So let's actually just quickly address what that means. So that's actually quite easy to calculate. What you do to calculate something called a mitotic index is simply calculate the number of cells that are dividing or that are in mitosis. So that means the 4 stages: prophase, metaphase, anaphase, and telophase. okay, over the total number of cells. That's the mitotic index. So if I have, for example, if I have an image on my screen and it has 40 cells, and I can see that 10 of them are going through the stages of mitosis, prophase, metaphase, anaphase, telophase, then my mitotic index would be 10 out of 40 or 1 out of 4. You can write it as a fraction. Most likely, you're probably just going to leave it as a decimal. That's my mitotic index. Very, very easy calculation. So for example, this is mitosis sample 1. What are the total number of cells observed in the sample? Okay, so if we take a look at mitosis sample 1, all right, so here it is. Okay, so the first questions, now it's probably best that you look at this on your own screen because it's maybe a little bit hard to see on mine, but it's going to be, the first question is, what are the total number of cells observed in the sample? Don't include cells where most of the cell is not visible. So you're going to have to go through this and you're going to have to maybe like just only include cells that you can clearly see all of the cell, all right? Or at least most of the cell. Like for example, I can see this cell right here, but this on the right side here, a lot of these cells are cut off, so I'm not going to include those, okay? So you're going to count the number of cells that you can see. And then in this sample, you know, the next question would be, in this case, how many cells show an intact nucleus? So that's where you would be able to actually identify a nucleus. So for example, in this middle cell, I can see a nucleus, right? But the cell above it, definitely no nucleus. So you're going to have to analyze the samples. If you have to do the mitotic index calculation, again, what you would do is you would look at the cells that are undergoing those 4 stages. So for example, if I look at this cell right here, on the left side, my second cell from the bottom, so bottom left corner, I can see prophase. All right, so that cell is clearly undergoing the stages of mitosis. Beside it to the right, so in the second column of cells, second from the bottom, I can see metaphase. So those 2 cells are undergoing mitosis. So what I would do is I would go through my image and I would count how many cells are going through those 4 stages, divide that by the total number of cells, and I would get my mitotic index, okay? So your job will be to go through the samples and answer the question. So the question that I actually skipped over was in relation to number 2. So, you know, for example, how many cells have an intact nucleus? And then the follow-up question is, what proportion of the life cycle does the cell spend having a nucleus? In other words, what that means is the same thing as the mitotic index. It's the same calculation because if I have, let's say on my slide set, I have 20 cells and 10 of them have a nucleus. Let's say 10 cells have a nucleus and I see 20 cells in total, then the calculation for number 3 is exactly the same as the calculation for the mitotic index. It's a number of cells over the total number, right? So in this case, it would be, I'm not saying this is the answer, by the way. I'm just giving you an example that this would be 0.5. So the way you calculate number 3 is exactly the same way that you calculate number 4, okay? And then you answer the follow-up questions. Now for mitosis sample number 2, some of the questions will be the same, some will not though. So make sure that you answer the questions that are in regards to that specific sample. So sample 2, if I go back, all right, so I look for sample 2. Here's sample 2. You can clearly see sample 2 is a different set, all right? Now, the questions for sample 2 are different then. It says, what are the total number of cells observed in the sample? So it can't be the same answer as the one before. How many cells are actively undergoing mitosis? That's the same. Calculate the mitotic index. That's the same. You know, so there are similar questions, but some of the questions are different. All right. So don't assume that they're always the same question. So make sure that you read the questions. Like for example, if I look at mitosis sample 3, I can clearly see that the questions are different than for 1 and 2. Okay. So your job is to go through all the samples and then answer all the questions. All right. So there's a few, you know, that you're gonna have to do. I mean, it shouldn't take too long, but make sure you go through them. Take your time. Might be a little bit tedious because, you know, counting the number of cells on this image might make you want to pull your hair out. Now, I don't have that problem, but you might. So I don't have it here. You know, so just take your time and just answer the questions, all right? In terms of the complexity of the lab, it's not that complex, but just take your time with it, all right? Any questions about this? So just make sure you match up the right question. So like it says onion skin versus onion root tip. So you're going to compare these two samples. So make sure for that one, you look at the right samples, okay? So onion skin is labeled onion skin. Onion root tip is labeled onion root tip. So in that case, you're comparing those two samples, all right? There is an example below it. So the second last thing on this, right above where you submit your lab, is called onion mitosis example. So, you know, that's where you would look for examples in case you forget, right? You can clearly see some examples of mitosis, all right? Okay. Any questions about the lab? Remember, I'm recording this in case you forget anything. You can always just go back. All right. Any questions then about this, girls? Okay. So then, the second thing we're going to do today, it's just hopefully going to be fun, maybe, is we're going to review. Oh, there's a question. Well then, when is it due? So let's say 2 weeks from now. Let's say not next week, but the week after. Okay, it's more than enough time, you know. But you know, we're not so, you know, given you know, how I don't know if everyone's going to have the same access to internet, computer, stuff like that. You know, I need to give people a little bit extra time. Okay, so I think 2 weeks is more than enough time. All right. Okay, so let's try the Kahoot. All right. So here it is. Let's try it. If it doesn't work for you, if we can't, then we'll just use it as a way to review. All right, review. Because we've covered 3 things so far. We've covered the intro to the cells. We've done cell specializations, and we've looked at cell division, right? So this is based on those 3 topics. So what's this? No, what do you mean sign? Just play it. All right, let's do this. Let's try this. Okay. Let's try this. So can you ladies see the Kahoot? You can see it? Okay. Let's see. Yeah, okay, good. Very good. Okay. So you can, if you're on a computer, you can obviously do like a split screen where you can set up the image of the Kahoot and the image of I guess, oh, I see some of you guys are getting in there. Okay, good. All right. If you can't get in, you can't set up your screen so you can see the questions. Just We'll just use it as a way to review, okay? But let's see how this goes. This might be interesting. Let's give us time, a little bit time. Can you hear the music? Oh, no? Oh, that's too bad. Really? Huh. Ah, that's no good. I wonder why you can't hear the music. Let's see. Let's see, hold on. Let's see if you can hear it. Hold on. Oh, sorry, just try something here. Now? Okay, so you can hear it now. Okay, that's interesting. So if I have my headphones plugged in, you can't hear it. Okay. So if you can, so it must be because if I have my headphones plugged in, you can't hear it. But if headphones are not in there, then I guess you can hear it. Okay. We'll just give it one more minute, and then we'll start. Okay. And again, if you can't get in, I apologize. If it's not easy, if you're on, like for example, if you're just using your phone for this, it would be very difficult to see the questions, and because you need to be able to see the questions and the answers together. That might be hard. You can set up a split screen on your computer. That'd be great. But otherwise, or the other option is if you have a computer and a phone, you can have the questions on the computer and you can answer on your phone or vice versa. So we'll start in a little bit. Oops. Sorry. It's not showing. Okay. E. Okay, I think we're going to start. And like I said, if you can't get in, you can just play along virtually, I guess, like you are anyways, and just use it as a review. So, okay, let's try. Ready? Here we go. Are you ready? Which of the following is not part of the cell theory? That's it. Not all cells have a nucleus. Not all cells have a nucleus. The other three statements are part of the cell theory. All right. So, next question. Well, D's in the lead. All right. Yes, that's right. All cells. So, not all cells have a nucleus. Not all cells have a cell wall. And bacteria don't have mitochondria. So, the best answer, cell membrane. Very good. All right. Next question. Here we go. Very good. Ribosomes make proteins that are made from the nucleoli in the nucleus. Excellent. Next question. Hey, it's in third place. Uh-oh. Talked about this. Think about the webbing between your fingers, or the lack thereof. All right, lysosomes. Okay, next question. Here we go. What are bacteria lacking? A nucleus. Very good. All right, they don't have a nucleus. Excellent. All right, next question. D is leading still. Remember that video? The inner life of a cell. Shipping and receiving. Purolator, Canada Post. Those are hints. Golgi complex. Excellent. All right. Next, we have a new leader. Hey. Okay, this disease, rapid aging. Terrible disease. Caused by defect in which part of the cell? Give you a hint, bacteria would not have this. Actually, that doesn't help. Bacteria don't have any of these except for a cell wall, actually. Nuclear membrane, very good. All right. Hayes in lead. Chem SC, Florence. Number 8. Which of the following is not a role of the cytoskeleton? Yes, detoxify. That's part of the smoothie R, and the peroxisomes are involved in detoxification. Very good. All right, hey, still in the lead here. Tough round. Okay, when there's not enough cell death, that's what apoptosis means. What could this lead to? Not enough cell death. Not enough cell death means too much cell division. That's a hint. Uh-oh, cancer. Too much cell division, right? That's too much cell division would be cancer. All right, next question. That was a tough one. Next question. Oh, Florence, look out, new leader. Uh-oh, we talked about this one just a little moment ago. They're involved in detoxification, just like the SER. Very good. Uh-oh, Florence, what happened? Next. In the middle of this image. I think you've just seen this one. Anaphase, very good. Chromosomes are split. Excellent. All right, Hayes back. Next question. What could this be? I don't see any chromosomes. Very good. Can't see the chromosomes, must be an interphase. Okay. Next question. Those are erythrocytes, red blood cells. Mammalian red blood cells specifically, by the way. All of the above. That's right. They don't have a nucleus, they don't have mitochondria, and they are round and thin. Look out. Here we go. Next question. Think brain. Neurons. Very good. Clara's on fire, apparently. What is it? Or what is it called? Yes, very good. Mitochondria is plural. Mitochondrion is singular. Excellent. Powerhouse of the cell. Uh-huh. What do plants have and animals don't? That's right, cell wall. Very good. Very good. All right, next. Okay. Next question. This cell is in what phase? Metaphase. Very good. The one got fooled by my middle phase choice. Very good. Okay, next. Oh, is in the lead. This cell is in. What can I see? I actually can see some stuff in there. I can see chromosomes. Prophase. Very good. Next. Cell in the middle. Looks like there's a cell plate forming. Telophase, very good. You can see that cell plate form, which means it's at the end of mitosis. Next, question 20. Is it A, B, C, or D? Look at the diagram. Which structure is the nucleolus? Not the nucleus, the nucleolus. This is the structure that gives rise to the ribosomes that read the RNA to make protein. All right, very good. That was A, right? That big central dark spot. There you go. Okay, next question. Which structure allows the DNA copies, which are called RNA, to exit the nucleus? Remember, it was in that video, Inner Life of a Cell. It came out of little holes. Do you see the little holes? Very good. Excellent. Okay. The nuclear pores, right? Nuclear pores. Excellent. Okay. Next. This process is called see chromosomes. See red and blue ones look like they're swapping something. Must be swapping DNA. This is called crossing over. Very good. That mixes up the DNA, makes new combinations. How do we get lots of variety for sexual reproduction? All of the above. All those things contribute to increased variety, including crossing over, which was the previous question. All right, next. Question 24. Compare meiosis and mitosis. What is different? That's right. The daughter cells have half the DNA because the sex cells, remember, have half the DNA, and they recombine to form the zygotes. Next, number 25. Roots of plants underground. There's no light, or very little light. Probably no light. Yeah, that's right. Chloroplasts, because they need sunlight to work, so it wouldn't make sense for them to be in the roots. Next. Read this carefully. Yeah, very good. Viruses are not cells, so they wouldn't have any cell parts. Next. The cell membrane. What is false? Yeah, it's not smooth. Remember, it has a rough appearance. All right. Next question. Plants and animals. That's right. Plant cells have a large vacuole, but animal cells do not. Everything else they have in common, right? Next. Hepatocytes are liver cells. But you don't need to know that to answer this question. Number of specialized cells. What makes cells different from one another? Yeah, it's their genes. Their DNA is pretty much the same. It's the genes that are being used. That's what makes them different. Okay, I think there's 2 questions left. Think about this. If you can't remember the term, you should be able to narrow it down to at least 2 choices. Yeah, so it can't be specialized cells because if you remember, specialized cells form later. The early cells are stem cells, and totipotent stem cells can be any type of cell. So that was a tough one. Okay, the last question I believe is worth double the value of all the other questions. So here we go. Oh, times 2. Remember we talked about this? When you get old? And the old feels new again. Wow, that actually went pretty well. And the winner is Cam SC. Clara in second, and up in third. Hey, and Asia. Is it Asia? Is that how you say it? Number 4 and 5. Very good. Excellent. All right, girls. That was actually a good way to review the cells, specialized cell, and cell division. So let me stop the record.